Long before a patient feels an ache, a lump, or a cough, a cellular error has already been multiplying in the dark. These microscopic masses expand silently, completely unnoticed by the person carrying them. In standard oncology practice, doctors have to wait for that mass to cause physical symptoms or appear on a scan before they can perform a tissue biopsy to see what they are dealing with. That waiting period forces medicine into a corner. By the time a surgeon can physically extract a sample of the tumor, the disease is already deeply entrenched in the surrounding tissue. Defeating cancer requires moving ahead of that curve. We have to intercept the disease at the microscopic level, well before it grows large enough to be seen. Researchers have found a way to bridge that gap. Instead of waiting for a tumor to announce itself, they are looking for its earliest traces inside the patient's own bloodstream. As cancer cells expand and die off, they undergo a natural decay process. When they break apart, they shed tiny fragments of their genetic material directly into the blood. Scientists call this material circulating tumor DNA, or ctDNA. The human circulatory system effectively acts as a real-time genetic ticker tape. Along with oxygen and nutrients, it carries specific, readable strands of code breaking off from the tumor wall. To read that ticker tape, clinicians only need a standard, non-invasive blood draw to send to the laboratory. But the blood is crowded with a massive pool of floating genetic material called cell-free DNA. The vast majority of this comes from perfectly normal, healthy cells constantly turning over. Spotting the mutated cancer DNA within that healthy pool is incredibly difficult. In early stages, a tumor's genetic signature is just 0.01% of cell-free DNA. Finding this requires next-generation sequencing to scan millions of fragments line by line for structural errors. Algorithms then act as a digital sieve, filtering out harmless age-related mutations to isolate the true cancer signal. The software algorithms filtering the data are as critical to the process as the physical needle used to draw the blood. Without them, the true biological signal remains lost in the noise. Once isolated by the algorithms, the mutation data goes back to the oncology clinic. The sequence data forms a comprehensive molecular profile of a tumor that hasn't even been located on a scan yet. It reveals the exact cancer type, how advanced the disease is, and its specific genetic driver mutations. With that precise intelligence, oncologists can deploy highly targeted, personalized therapies designed for that specific genetic flaw, bypassing the blunt systemic approach of generic chemotherapy. This chart shows how clinical trials actually map patients to these specialized treatments. Looking closely at Design 4, you can see how a positive ctDNA blood test leads directly to an experimental treatment pathway based entirely on their specific blood results. The blood draw reveals the enemy's exact molecular blueprint, giving doctors the precise coordinates they need to destroy it. This approach upends traditional cancer care. We are moving away from late-stage crisis management. Instead, oncology is shifting toward real-time, proactive surveillance of the body's internal state. In the near future, this complex sequencing technology will be as routine and accessible as checking your cholesterol during an annual physical. The ultimate goal is a single, simple blood test capable of catching multiple different types of cancers simultaneously. This means catching a tumor at its most vulnerable, microscopic stage, months or even years before the first physical symptom appears. Liquid biopsies rewrite the entire timeline of the disease. By intercepting cancer before it takes hold, they buy patients the single most valuable resource in all of medicine: time.